Hi everyone. For a long time, the conventional view of myelin has been pretty straightforward, a passive insulator for the brain's wiring. But as it turns out, there's a growing body of evidence that suggests a much more dynamic and frankly, a much more critical role. So today we're going to walk through a comprehensive scientific review by Huang and colleagues. This review really reframes our understanding of myelin's function, details its breakdown in a wide range of central nervous system disorders, and then outlines some of the emerging therapeutic strategies that are designed to promote its repair.
So to structure our discussion, here's our roadmap. We'll start with the fundamentals of myelin biology and its capacity for plasticity. From there, we'll examine its diverse functional roles. Then we'll get into the pathology, investigating how its dysfunction contributes to aging, Alzheimer's, and other conditions. After that, we'll review a class of pro-myelinating compounds and wrap up by discussing the strategic challenges and future directions for remyelination therapies.
All right, let's begin with the fundamentals. To really understand how to repair myelin, we first have to have a solid grasp on how it's constructed by its specialized glial cells in the first place.
Okay, so the cells responsible for producing myelin in the central nervous system are the oligodendrocytes. Now these cells, they develop from a population of progenitors known as oligodendrocyte precursor cells, or OPCs. These OPCs are generated in specific neurogenic niches, and then they migrate pretty extensively throughout the CNS. From there, they undergo this multi-stage differentiation process, maturing into cells that are capable of extending these long processes that then wrap around axons to form that compact, multi-layered myelin sheath.
Now, it's really crucial to understand that myelination isn't a static, one-time event. We can actually distinguish between two fundamental modes. On one hand, you have innate myelination. This is the developmentally programmed process that establishes the bulk of the brain's wiring early on in life. But on the other hand, you have adaptive myelination, and this is a lifelong process. Neuronal activity that's driven by learning, by acquiring motor skills, even sensory experience, all of this can trigger the formation of new myelin sheaths. What this does is it identifies myelin not as some kind of fixed infrastructure, but as a key substrate of neuroplasticity.
So, if myelin is more than just insulation, what exactly are its functions? Well, its contributions to the physiology of the central nervous system are proving to be far more extensive than we previously understood.
This schematic here gives us a good summary of 4 key functions of myelin. These include facilitating rapid nerve impulse conduction, providing metabolic energy to axons, contributing to learning and memory, and potentially serving as a lipid-based energy depot. Let's take a look at each of these in a bit more detail.
Myelin's best-known role, of course, is enabling rapid saltatory conduction. By restricting ion channels to the unmyelinated gaps, the nodes of Ranvier, it increases nerve impulse velocity by as much as 100-fold. But beyond just speed, it provides critical metabolic support. Oligodendrocytes supply axons with energy-rich metabolites, things like lactate and pyruvate, through a whole network of transporters. We also now understand that this adaptive myelination is a mechanism for consolidating memory and refining motor skills. And finally, there's an emerging hypothesis that suggests the vast reservoir of lipids within myelin might actually serve as an energy source for the brain during periods of high metabolic demand or scarcity.
So given these critical functions, it follows that the failure or loss of myelin would have pretty severe pathological consequences. We're now going to turn to myelin dysfunction, and we'll start with its role in normative aging and in Alzheimer's disease.
As this diagram illustrates, compromised myelin integrity isn't some kind of niche pathology. It's actually a common feature across a remarkably broad spectrum of disorders, everything from neurodegenerative and psychiatric conditions to traumatic injury. This suggests that myelin pathology might be a convergent mechanism that underlies the symptoms of many distinct neurological diseases.
In both aging and Alzheimer's disease, the evidence for myelin dysfunction is quite compelling. Studies in aged mice show a decline in oligodendrocytes and in myelin renewal. In humans, imaging studies correlate this age-related myelin decline with reduced memory performance. Now, for Alzheimer's, evidence from mouse models suggests myelin loss is actually an early event, even preceding other classical pathologies. And of note, the APOE4 allele, which is the primary genetic risk factor for late-onset AD, appears to directly disrupt myelination by interfering with cholesterol transport. These findings are then validated in postmortem human brains, which show extensive myelin degradation.
This quote from the review highlights a finding of considerable significance. In mouse models of Alzheimer's, genetically stimulating the formation of new myelin was sufficient to rescue memory deficits. This occurred even in the presence of established pathologies like amyloid beta plaques and microgliosis, which is inflammation driven by microglial cells. Now, the implication of this is quite significant. It suggests that targeting myelin repair could be a viable therapeutic strategy for Alzheimer's that operates independently of the amyloid cascade, which, as you know, has been the dominant focus of drug development for decades.
The impact of myelin pathology extends well beyond aging and Alzheimer's. So let's briefly examine its involvement in multiple sclerosis, major depression, schizophrenia, and ischemic stroke.
Multiple sclerosis is, of course, the canonical demyelinating disease of the central nervous system. This pathology is recapitulated in experimental autoimmune encephalomyelitis, or EAE, which is the principal animal model used to study MS. A key feature of the human disease is the failure of endogenous remyelination. OPCs are often present right at the edges of lesions, but they fail to differentiate and repair the damage. This seems to be due to a combination of inhibitory molecules in the lesion microenvironment and epigenetic blocks within the OPCs themselves.
There are also strong links to psychiatric disorders. In animal models of depression, for instance, chronic stress induces myelin loss in key emotional regulation circuits, and restoring that myelin can actually reverse depressive-like behaviors. In schizophrenia, postmortem and imaging studies point to deficits in white matter integrity. Animal models have specified this even further, showing hypomyelination of critical inhibitory neurons, like parvalbumin-positive interneurons, which could disrupt the precise cortical synchrony required for normal cognitive function.
Ischemic stroke also causes significant myelin damage. Oligodendrocytes are extremely vulnerable to oxygen and glucose deprivation, and this leads to widespread and persistent demyelination in the affected territory. Subsequent research has demonstrated the functional importance of repair. In animal models, blocking the brain's own attempts at remyelination after a stroke actually worsens the long-term motor and memory outcome.
So, given this widespread evidence of myelin pathology, a key therapeutic goal is to identify compounds that can stimulate the brain's innate capacity for repair, and high-throughput screening efforts have now yielded several promising candidates. This table highlights several compounds that have been identified as potent promoters of oligodendrocyte differentiation. One prominent class, which includes repurposed drugs like clemastine and benzotropine, acts by antagonizing muscarinic acetylcholine receptors. However, other compounds function through entirely distinct pathways, targeting intracellular signaling cascades, nuclear receptors, or even epigenetic mechanisms. This mechanistic diversity is quite encouraging, as it suggests multiple points of intervention are available to promote remyelination.
We can think about these strategies in two main ways. The muscarinic antagonist approach is essentially about releasing the brake. OPCs express these M1 and M3 muscarinic receptors that act as an endogenous inhibitory signal, preventing them from differentiating. So by blocking these receptors, these drugs remove that stop signal. In contrast, other compounds work by pressing the accelerator, actively engaging pro-differentiation pathways through intracellular signaling or by overcoming the epigenetic silencing that can accumulate with age. Identifying these compounds represents a major advance. However, translating these preclinical discoveries into effective therapies for human patients presents a formidable set of challenges. So, let's consider the strategic framework that's required to make remyelination a clinical reality.
Successful remyelination is unlikely to be achieved just by targeting a single process. As this framework illustrates, a comprehensive strategy has to incorporate three components. First, you have to limit the ongoing demyelination. Second, you have to promote the efficient clearance of inhibitory myelin debris. And third, you have to directly stimulate OPC differentiation and new myelin formation. These are not independent goals. They're actually interdependent requirements for functional repair.
So the first priority has to be to control the underlying pathology. Promoting the formation of new myelin is of little use if it's immediately destroyed by the same process that caused the initial damage. For a condition like MS, this means concurrent immunomodulatory therapy. In the context of aging, this involves addressing the chronic, low-grade inflammatory state of the brain. This all points to an eventual need for combination therapies that can both protect existing myelin and promote its regeneration.
The second strategy is to manage the aftermath of demyelination. You see, the lipid-rich debris that's left behind isn't inert. It's pro-inflammatory, and it actively inhibits the differentiation of OPCs. So, efficient clearance of this debris by phagocytic cells like microglia is a prerequisite for repair. Therefore, therapies must enhance this cleanup process, ensuring the phagocytes clear debris effectively without inadvertently targeting healthy myelin, a process that requires tightly regulated intercellular communication.
And finally, the third strategy concerns the pro-myelinating compounds themselves. Several critical pharmacological hurdles exist. For one, the drug has to be able to cross the blood-brain barrier in sufficient concentrations.
Off-target effects have to be carefully evaluated, particularly for receptors that are widely expressed in the brain. And most importantly, the impact of age has to be considered, because OPCs in the aged brain exhibit cellular senescence and may be less responsive to pro-myelinating signals. This could potentially limit therapeutic efficacy in older populations.
In summary, the evidence compellingly reframes myelin as a dynamic and indispensable component of CNS function. Its failure is a central pathological feature across a wide array of neurological disorders. And this leads to the ultimate question: what is the key to unlocking functional myelin repair? The research suggests there isn't going to be a single solution. The answer will likely involve integrated, multi-pronged therapeutic strategies that are tailored to the specific pathological context of each disease, combining approaches that limit ongoing damage, clear inhibitory debris, and enhance the brain's own remarkable capacity for regeneration.